Hello, everyone. We're just giving a couple of minutes of courtesy for people that might be running a couple of minutes late. So we will get started in, again, about 2 minutes. One more minute, and then we get started. We're just making sure that everyone or most of the people that registered can actually make it. Okay, everyone. I think we can now get started. It's a great pleasure to have you all here today. We're going to be talking about AION, this AI-based tool for clinical variant interpretation. This is a tool that is developed by Nostos Genomics. Nostos Genomics, we are a German company headquartered in Berlin. We have people working across Europe. And our expertise lies in AI and developing these type of tools to streamline genetic analysis, especially for rare diseases in clinical setting, but also in research setting. So today we're going to do a live demo. This is a brief agenda for today. I'm going to introduce to you AION and the reason why we are building it, and we've built it and keep improving it every day. Then we're going to go into a demo, and then we have a section for Q&A. I'm your host. I'm Pablo Botas. I'm the head of product here at Nostos. And we have with us the great Aina. Aina is the clinical customer success manager, and she is going to be in charge of running the demo and also the last part of the slides. We're going to try to make it quick. We're going to try to focus on the why we do what we do and what you can achieve with AION, and then obviously the demo will dig a little bit deeper on the how you do things with AION, but we'll try to keep on the why and on the what of things, because that's what actually drives us as a company, and I think it's also what can make a change in the industry that we're focusing in. Before getting started, I want to share a bit about the QA. During this demo with Zoom, you have the QA functionality activated. So at any point, you can ask questions through the QA, and then we will try to get back to them as soon as possible, most likely already after the demo is done. So make sure to stick around until then, because that's where the most interesting conversation or the closer to a conversation that we will have, and that's going to be very, very interesting. So as I mentioned, I'm going to start a little bit on the why we do what we do. We are a company with a clear vision, and we want to provide answers to patients and basically empower them to make better healthcare decisions for themselves. And we're very sensitive to the topic of rare diseases. Some of the founders have personal experience with rare diseases, with very close family members, but they are a worldwide problem. Rare diseases in general, they affect more than 350 million people worldwide. Some estimates give about 400, it depends on how you count them. And there are more than 7,000 rare diseases. Again, it depends on how you count them. Sometimes it goes up to 12,000. The problem with them is that they are difficult to diagnose because people don't know how to really understand them, how to really, first of all, suspect that there is a rare disease, then order a genetic test to try to provide an answer. But even the genetic tests take some time to be ordered, and then there is a big interpretation bottleneck in there. So access to genetic tests has been improving over the past years, but still there is an interpretation bottleneck there. And overall, the time that it takes for diagnosis varies depending on the geography, but it can be 6 to 8 years in many places. So as I was saying, genetic tests do provide answers in many cases, not in 100% of them, sadly, but they do provide answers. The problem is that the access for them has been scaling, but not so much the efficiency and the capacity of the market. So we wanted to address this bottleneck. Basically, the bottleneck looks like the following. You have a number of input variants, and analysts within genetic testing labs, they need to go through them and identify the one or the ones that actually can explain the patient disease. And this is no easy task. This is a complicated process that takes many hours. In this case, we're highlighting one for whole genome sequencing, which can be a bit of an extreme case where an average can be around 10 to 16 hours, depending on the tool that you use. For whole exome, can be about an hour, 2 hours, something like this. For smaller panels, the time might decrease. Nevertheless, this is a very laborious process that takes a lot of expertise to follow and takes a very long time to finalize, especially in whole genome, but this is not the case for most cases in the industry. As a consequence of this, analysis times are very long. They are spent, a lot of time is spent in cases that could be solved much, much faster if modern technology were applied. And again, as a consequence, only about 30% of the cases are diagnosed, with 70% of the cases remaining undiagnosed. This is a ballpark number, but it already shows and showcases the problem. What we have built is AION. This is an AI-driven solution that we're very happy and proud of. And basically, the goal is to point our users, who are these genetic analysts, to answers straight away. For that, we do phenotype-driven prioritization, and it has a lot of molecular insights as well. And Aina is going to show that in way more detail, so I'm not going to dwell too much on that. And basically, a solution might look like this. Look, for this case, the message that AION is sending is that there is this variant that looks really pathogenic because of this, and that. Aina will explain. And what we're trying to bring into the market is a tool that can really, really streamline this process. Streamlining this process means that you need to be able to predict what variants are actually pathogenic or disease causative. We have such a technology, and we have actually validated it independently with cases from Genomics England. These are cases that we had never seen before. Actually, the validation happened within the Genomics England infrastructure, not in ours. This is to say that we basically send our algorithms to them, and they run the algorithms on an independent cohort. The cohort was made out of patients that had a genetic disease from the 100,000 Genome Project. And basically we took as inputs, or they took as input, the genetic data and the clinical data for each of the cases, run all of these through AION, and then we analyze the results. Actually, these results are kind of outdated. We have a much better sensitivity now, but this is an independent study with independent cases. And already the results a couple of years ago showed more than 90% sensitivity. This means that AION is able to tell you the right answer and suggest you the right answer in more than 90% of the cases that otherwise you would be able to diagnose. So as an alternative to a very laborious process, you can simply feed all of this to AION, and it's going to pinpoint for you the right answers for a very quick evaluation. And this is basically what our customers are saying. This is Dr. Giuseppe Novelli from Tor Vergata, but this happens all across Europe where we have many customers. And basically what they are witnessing is that AION actually allows them to solve this bottleneck and focus on the hard cases. At the end of the day, it's all about giving better answers to the patients. So basically what AION is allowing them to do is to automate this variant classification, to take into account the phenotypic and clinical data as it should and take certain center stage, and all of this through a very user-friendly interface that allows people with maybe less expertise also to be highly performant. Beyond the performance, AION is a tool that adheres to the highest standards of trust. It's a CE-IVD medical device. We are actually expecting CE-IVDR by the end of this year. We don't think that it will go to beginning of 2025. I think it's going to happen already during this year. It obviously compliant with GDPR and also with ISOs 13485 and 15189. This is all very important. Basically, we're dealing with genetic data, and the platform is developed in a way that it's highly compliant and adheres to the highest standards of security and trust. So before moving on to the demo, I wanted to also take advantage of this opportunity and invite all of you to an upcoming webinar that we have. It's a webinar that is not going to talk about variant interpretation in itself. It's going to talk about labs, and it's going to talk about differentiation of labs and how labs can differentiate and provide better service for their customers. So we're going to be sharing a link to this webinar as well, but you can also scan the QR code here. You can also scan the QR code here very quickly, and you will be forwarded to the registration page. Okay, so with this introduction to the problem that we're trying to solve, I just want to give the floor to Aina to take us through the demo and a brief explanation at the beginning.
Hi, thank you, Pablo, for the kind introduction, and welcome everyone. So today I will take the opportunity to have the time to present you with like 2 cases showcasing the main features of our platform AION. So basically, I am going to show you the potential that our platform has to help you, as Pablo mentioned, to streamline your processes and be able to just decide on like how you want to review the cases and get diagnostics as soon as possible instead of having this bottleneck of hours before reaching a conclusion. So basically today in these different 2 cases, I will show you how AION can present like the variants that are present in your files in a ranked list so that you have a short list in which you can focus and find like the causative variant for your patient in a very fast manner. This way, we implement like the different resources like that are available in the clinical field, and also incorporating like ACMG classification automatically with our also like AION algorithm that will give you like the AION predictor score that I'm going to show you later as well. We base this prioritization also using like the phenotype that you input from the patient. So it's not like you have to go and deep into a very like long list of variants that it shows you like as kind of like an Excel file that you need to just filter through it. We just like give you like this prioritized list in which we consider the phenotype and let you go to the point. Also related to this phenotype, we do deep phenotyping, and also like by providing like the overlap from your patient with the symptoms associated with the disease that are associated with the variant we are seeing, it allows you to go back, review the symptoms, and see whether you were actually missing any like some symptoms from your patient, and be able to reach further conclusions on whether this variant that you are seeing is actually very relevant for your patient and include it. Also, something that AION also provides, we, of course, as I was mentioning, provide like all the necessary resources so that you can get like an informed decision and confidently report variants for a particular patient. But we also like allow to have this internal database. This internal variant database, what it allows you is like on top of like data such as like NOMAD frequency there, you will have like your own internal numbers, which give you like insights into your own population, and it will give you like some further insights on what is relevant for your particular patient. Also, AION supports any type of cases, as Pablo was mentioning. We support like panels, whole exome sequencing, whole genome sequencing, and also all type of variants, both like small variants, but also we include CMV prioritization, including like the ACMG criteria automatically applied as well. I'm gonna also show you this in a particular case. And also, I just want to highlight that aside from doing this like a variant ranking in a shortlist, we also allow to the users to play with advanced filtering. So we allow cascade filtering that you can save and share with your team members to have like a structured way of like filtering with different like logics and being able to filter by the most relevant criteria that is relevant for your case assessment. I'm also gonna go deep into that. And finally, in terms of like filtering, we know that there are like sometimes restrictions in like which genes or people can report. So we'll also be showing like you can also just restrict the number of variants that you are reporting by applying like in silico panels, and this can be done either before the submission of the case or after the case during the analysis, so that you only focus into the variants that you want to report. This also like includes like being able to just like exclude any secondary findings. Finally, as like a solution to provide like all the support to the clinicians and like researchers to solve clinical cases or research cases, we want to provide like a very fast and easy solution. So basically, we support you end to end. So from either FASTQ files or VCF files, and together with the HPO terms or symptoms they will input from the patient, we just like in minutes we will be able to provide you with the answers. So having said this, here you have a bit more in a graphical way how the process works. So as we said, like our platform supports any type of analysis. So here you have an example like from what the analysis would go through from like the VCF files, either for a single or a trio case, would just first like do the annotation of the files, classify the variants, and together with the input of the symptoms from your patient, we will highlight the variants that are more relevant clinically for your patient in this particular case. This way you will have like a short list, and you will be able to explore the variants that you will find more relevant in terms of pathogenicity, and like select your candidates to be able to report in your particular template to your patient. So this is like mainly like the process and how it goes. And just finally, to end this presentation, what I would like to share with you is all the specifics on the cases that we are going to see today. So I'm going to share my screen now with AION. Let me 1 second. Okay, so I think now you should be able to see my screen. So basically here, first of all, you see the first case I want to talk today. This is a trio case. you can see always in AION on the top side, the clinical information, so a summary of that with all the patient symptoms that was introduced at the time of submission. This is, as I said, like a trio case with unaffected parents. On the right-hand side, you have all the summary also with the analysis information, so which files were considered for the analysis, the reference genome version, and if any restrictions were done as gene panels applied at the time of submission. Also to highlight here, as I said, like we support both small variants. As you can see here, there are two first steps regarding small variants, and finally one for CMVs. The most relevant part to focus at the beginning is this short list of AION ranking variants. This is like a very short list that generally ranks from 10 to 15 variants that will give you the further, like the most relevant insights for your case. So it will like highlight you the variants that have like a most significant impact, considering like molecular aspects and clinical aspects, such as like, the impact of the variant, the frequency, the pathogenicity and the deleteriousness like scores, and also like considerations in terms of segregation in the case of like considering a trio case. So for example, in this particular case, you can always see that the variant contains all the relevant information. Starting from the top, you can see there's this like a score number, which brings you like a phenotypical overlap from your patient to the specific disease that is associated with this gene. When you want to see how this associated is done, you have always this information and always the links to the original source. Also like the definition of the disease and links to the OMIM and PubMed publications. Now, when we dive deeper, you can see here that you can see all the symptoms associated with the disease, and in darker gray highlighted the ones that are overlapping with your patient. It is also like relevant to highlight that we also always show like all the symptoms here, because like as I said before, like by showing that, and you can always explore a bit further and see whether there is like some symptoms that actually were missing to be reported in your patient. So for example, in this particular case, it's a real case in which so these are like the patient was, aside from the symptoms that we are seeing here highlighted in darker gray, after exploring like and asking the family, it was found to have like recurring infections. So this like, expanded like actually the phenotypical overlap, and also deeper looking, they also could associate with kyphoscoliosis and other like symptoms that are like were found later on as like some dysplasia in terms of like ossification. Also here, you can always see like the inheritance pattern, variant effect, and variant information. And on the bottom, as I was saying before, you will have like information in terms of like ClinVar submissions. If you have the AION database activated, any previous classifications you did for this variant, particular variant in other cases, which I will show in depth later, the automatic application of the ACMG criteria, and the score for AION predictor. Let me zoom out a little bit here, because we are seeing this first variant I was mentioning. If you see though, like in combination with the second one, when exploring this particular case, it was seen that actually the first and second variant combined. This is a case of compound heterozygous, in which this first variant inherited from the mother combined with the father were like actually causing the like symptomatology that was seen in the patient. And this was like a very nice case to see, because like this was actually an unsolved case, and after submitting in AION, these like variants were like ranked the first ones in this short list of variants. Also, just going briefly back with what I was saying, these two variants like combined, you can see here that in some of them, like based on like ACMG criteria, would have been like reported as BOOS, and there's no ClinVar submission. But after like applying our machine learning algorithm, the AION predictor score gives it as pathogenic, and like the second one that would have been like a BOOS, it's rescued by being a compound heterozygous. So always like a compound heterozygous would be reported together, so to facilitate the work of reporting for the clinician. The AION predictor, you hover over it, you can see that it's a result of like combining different data points, so more than 100 data points that will give you a score from 0% being leaning towards benign to 100% leaning towards totally pathogenic. This is as I said, a combination of multiple data points. And if we open here to show more details, you can explore a bit deeper on different subscores that contribute to this AION predictor score. So AION considers also at the level of the amino acid constraint score, also what is the conservation at the evolutionary level, regional level, and also considers splicing scores. But in going deeper as well, in terms of the impact of the variant, the pathogenicity and deleteriousness, you have here on the bottom other annotations that are considered, such as predictors as SIFT, polyphen, grantam, or blossom, in terms of more pathogenicity and deleteriousness, also conservation scores to see how it's conserved across evolution, and also information in terms of NOMAD. So we incorporate NOMAD information in NOMAD B3, but also B4. So if there's data there, it will show here in this particular variant. As we see here, there is no information, but in the second one, if it is contained, it will be always showing here on the bottom. So you can see here the NOMAD B3 information and B4. Something very important to highlight, which is very nice. So for people that is using still AG19, we did the effort to do a liftover. So they will also have the information for NOMAD B4 in their cases using AG19. So this is an extra information that can be useful for the clinicians. And aside from this information, if it's related with NMD splicing, you will also have the scoring here in this area. Showing this information, we can now go to the next step, which, as I said here before, for example, this is a BOS case in terms of classification. You can always see how the automatized ACMG guidelines are applied. So basically, AI selects the relevant guidelines, but because we know there's some flexibility in terms of how in some particular cases or diseases or variants the criteria are applied, we allow the person, the user, to select different criteria if you have some interpretations on how you want to apply the criteria. So I'm just selecting one just to put the example, but when you select it, it will change here on the top. So your input is going to be considered on it's going to be recalculated. So you can always change that. And AION will always be transparent which criteria is being applied to consider this final classification for ACMG. Then you always have the information of quality, literature and resources. So basically PubMed publications, a link to a meme again, UCSC browser, publications in Mastermind available. And something very relevant that I was mentioning before, you will always have the classifications that you have been provided to this particular variant in other cases and the statistics of this particular variant in your own database. So including the allele count, the samples that contain the variants incorporated in the whole cohort in your database, and the frequency of the variant, among others. In terms of the variant classification, you can always click here to show the details, and it's very nice to be able to click and show the whole context in where this particular variant was classified. So you know the case, the person, date, and the information in the context in which this variant was classified, and any comments. So you can easily use this variant to this classification to apply into your particular case, or you can manually again classify the variant and write your own comments. After you write your comments here, you can save them, and then we'll show in the user interface. So if we go back now to the variant, it will show here on the top. So this is very useful, to incorporate into your own classifications, and also this particular classification will be incorporated in future cases, so you can have an additional insight for this particular variant. One more thing for all of the variants, you can see here, as I was saying before, in terms of variant information, to have even more reassurance on the variant, you can always visualize it in IGB. So if you have open IGB desktop with your BAM files, you can additionally visualize the variant there, as well as in UCSC browser. In terms of the AION database, as well here in this section, aside from having the details in show more details, you can always have the statistics here inside. So this is most of the information that you can extract from this particular vision. The only other thing that I wanted to mention is that, as we were saying before, when you review a particular variant and like the symptoms that overlap with your patient, if you have additional findings, something that is very nice in AION is that you can always edit the symptoms and incorporate any additional symptoms that you might have found in your patient, select, and it will be incorporated here in the list of symptoms, so you can always like save and resubmit the case. If you do that, everything is going to be recalculated because the platform is going to be considering the new input that is used in your case. And so that it's going to be bring up like the most relevant variants for your particular case. And it will always be like recorded here as version. So everything is going to be tracked. Having said that, once you are like ready to take a decision in a particular case, you can just like select the variants you want to report and write a case summary. So you will write here a report summary with like the relevant information you want to report on the case, and you can download the report. You can also equally like download the data and use your own templates, but this would be the main process that you would be doing when analyzing a particular case. And then also, like as we were showing, like the AION is giving you a very short list, but you can always like additional filters and be like filtering by genes and other parameters such as like inheritance pattern, zygosity, etc., based on like your own requirements. Now, something I want to briefly highlight is that the usefulness of the AION ranking, because this first step, as we are seeing like only 10, 11 variants in this particular case for this, patient. But here we see like actually we would have started with like almost 52,000 variants. That's all the variants that were annotated with this case. However, sometimes like, or because of like some historical like ways of like analyzing cases, you might want to use advanced filters. So play with this long list of like annotated variants, and instead of like going to this first like tab after like reviewing what information is there, you can also like be willing to play with filters. So what you can do is like click in here and you can create your own filters that you can even like share with the whole team. And as I said before, you can have like a structured way of filtering and a consistent way to analyze a case. So you can do it in different ways. So you can, for example, like, have like a specific set of genes that you would like to filter by. You can either like have them like searching here or like paste them here separated by a comma. So you can, have this set of genes, then also combine with different criteria you want to be filtering. So for example, you want to select genes variants that based on ACMG criteria are only the pathogenic ones. And this you can keep like adding criteria until you're satisfied. So maybe, you will also like want to have like variants that have phenotypical overlap with your patient, and you can keep going with combining with criteria of like logics of and or. So after you are saving the filter, you will have it here and you can apply it. So I have like some particular ones that I have made on my own criteria, which is like this ones, which is like considering like a CNG classification, clipper submissions, classification, phenotypical overlap, and frequency in normal. So for example, I can filter that, and instead of having this 53,000, now I have 391, which makes like my life slightly easier, but also you can, keep filtering by different criteria such as consequence, allele depth, etc. And you can be more or less stringent, and this will like keep reducing the number of variants you're seeing here. And by default, they are like sorted by position, but you can use different criteria, such as like you might want to, for example, like order them by phenotypical match. So having like the ones that show high overlap with the patient at the top. So you can be playing with the data and end up like with a similar solution that you can like, find here with AION. But of course, it's going to take you like much longer time because you need to play with the data and like, define like your own criteria. So this is all like for what you can show you for like the small variants. Now I just want to go briefly to this other case in which I'll show you the CMBs. So in the last step of a trio case in which you submit both small variants and CMB files, you will see that here you have this like table in which it contains a rank list of CMB and structural variants. All of them have comprehensive information, and you will see also, you'll notice that there are two subtabs. The first one show the relevant variants, and what do we like consider a relevant variant. A relevant variant is the ones that are considered like, to have an important like impact in terms of pathogenicity. So basically all those that according to ACMG guideline classifications would be pathogenic or POS, pathogenic, likely pathogenic or POS, have a low frequency, and also this is optimized by length. So if we click one of the variants, you can expand here, you'll see like whole information, seeing like this particular variant, for example, contains 7 genes in both the ones that are associated with disease, all the whole information in terms of the variant, and also information whether this variant has been, reported in other like established databases, such as here, for example, DBBAR, and you can go to the original source. We not only, of course, like have DBBAR, but like many others, such as like Decipher, DGB, 100 Genome, CMRI. So if there's any known reported CMB that overlaps with the particular region in which the CMB in your patient is found, it will be reported here. And also here you have the ACMG guidelines applied that you can go with more details here. So basically you can see that it gets like a 1.15 score by combining 2A and 5H criteria. And if you want to see a bit more in terms of like the DBBAR classification, you can always just go to see more details. On the bottom of every card here, you will also find like all the affected genes contained in the CMB. And when expanding, you'll find on the top the gene and the link to OMIM for this particular gene. If there's any information in terms of dosage, so here you have like the information of haploinsufficiency score being 3. With here you always have the tooltip explaining like the meaning if you're not that familiar, and the link towards like the ClinGen source. This like gene is totally contained in the CMB, this is the location, and then you have this is associated with this particular gene, inheritance pattern. And if you click, you can see what are the symptoms overlap with your patient. Also on the bottom of every gene, you will find like if there is any additional scores and predictors, such as like scores like and predictors PLI, LEF, and if there's information in P-haplo and P-triplo, you will also find it here. So this is like mainly what it contains, like a CMB card. And this is like going to be the same for all the genes contained there. What I can also show you is that, of course, in this particular case, you have 22 results, but again, like the small variant, sorry, you can apply and generate and apply filters. So for example, if we only like limit the CMB that we are seeing by ACMG guidelines being pathogenic, having some phenotypical match with my patient, and having been seen as pathogenic or no other like database classifications on this particular CMB, we can reduce this 22 variants to much slower number. So basically it's 2 results, and it makes basically your life to solve this case much faster. In this particular case, actually the variant rank number 1, so this CMB was the causative variant for this particular patient. So basically AION again, like, showed a great performance into, helping you to solve the case. And just to mention though, like here we were talking about the relevant variants. If we move towards like all variants, this is like all the annotated CMBs that you would find. So based on our advanced filter, now we are seeing 8 results. But without that, when you explore all variants for CMBs, you would be seeing over 5,000 results, which is like incrementing the effort you need to make for solving a case. So basically, this is like all you can see in terms of like CMBs. Again, like, you can, whenever you like, explore by yourself, like the platform, you will like be able to explore all the tables, all the information. But basically here you have all the variants ranked based on the criteria I was providing you. So I think this should be everything for today as a brief introduction. I think like now I can hand it back to you, Pablo, and we can go to any questions that we might have from the audience.
Okay. Thanks a lot, Aina. I always love seeing these demos. It's like seeing a baby in action. Okay. We have some questions in the Q&A. Please keep adding them if you still have some of them. I'm going to read through some of them, and again, some of them I might respond directly. Others I'm going to let Aina go through them. The first one that I have is it just VCF files that I can upload, or can I add FASTQ files? And Aina, please.
Yeah, sure. So basically, as I was mentioning briefly before entering to the demo, we both support like from FASTQ files or BCF files, so we can provide secondary and tertiary analysis support, but also like tertiary only. So basically, it's very flexible depending on your needs.
Okay. Again, if you have any other, keep adding to the Q&A. Next one, it's well, thanks for the nice presentation. Are only HPO terms available for entering clinical terms, or is there also a free text option? For some rare diseases, HPOs are not well defined. And I'm going to couple this with one that is about, does AION work without HPO terms? I think those two could be coupled together. If that's okay, I can take these two.
Sure.
First
Yeah.
of all, yes, AION does work without HPO terms. Simply that the specificity is a little bit smaller. The sensitivity is not affected at all. So the sensitivity is not affected at all. Sorry, I got distracted by your question. You can at any moment add phenotypic information during the analysis, as Aina shown. Then on HPOs available for some diseases, I think that's a very good point. And some diseases are not well described in the literature yet on the basis of HPO terms. In principle, one shouldn't be limited by that. The phenotypic comparison algorithm that we have is pretty robust for using HPOs that are not very well determined, meaning they're not very specific, meaning they're not like the last items in each of the branches. So in principle, AION is relatively robust towards that. It is true, obviously, that if a disease is not well described based on HPO terms, the specificity might drop a little bit, but because we show the direct comparison between diseases and the patient phenotype, if you have more information, if you're an expert in some of these diseases, you can also take that information into account and take that into account for interpretation. This is not a common situation in clinical genetic testing. It is more common in research. And for research, we do have some other functionality that is very useful. Talking about research, another question that we have is, I'm a researcher interested in this, I guess, in AION for processing all cases and harmonizing annotations. How could I use AION for this? Is it useful? Can I analyze multiple cases? Aina, do you want to take it? You're muted.
Sorry, I was going to say like I couldn't hear it very well the first part, but I think like you were mentioning about like analyzing multiple cases, right, in AION. So basically,
yes, for research purposes, basically.
So basically, yes. So AION allows like, batch launching. Basically, you can define like in a TSV file what are the requirements of your case, and then like you can just like launch the cases in batch, and then therefore like you will minimize the manual effort for launching. Yes.
Great. We have actually the last question that was asked. I'm not following the order of when the question were asked. What is the difference between your tool and Marvel? Did you do any comparison of that? And to be honest, I'm not familiar with Marvel. I can look into it.
I know, like, so basically Marble, like, so Marble Tour, what I know and I've been reading is like there's this recent publication in 2024 about this tool. I mean, it seems it's pretty nice. I still like, we didn't do any particular comparison. However, also there has like a number of limitations, which is also like a reason that we didn't compare, which is, for example, they do not support CMBs. So this is like obviously like, you know, a strong limitation, and like because of course like you know, you cannot be only limited into small variants. So this is why like at the moment now we did not like consider doing this kind of comparison. But so basically I cannot touch a lot onto that. I know like has been like a recent like a tool that has been come out and there's a publication about it. But yeah, for the moment now we would need to just like keep exploring further. And if there's particular reason or areas that you would be interested in exploring or knowing the, you know, the details in AION, or like, you know, to compare the utility for your particular cases, we can deeper talk about this.
Yes, having a quick look into it, it seems that it's a research use only tool, whereas AION, I think it has more functionality. It allows you to do more things with especially clinical cases, but also for research applications. I cannot really comment on how annotation works and how deep it is between the two tools, but definitely AION lives within an environment that is CE-IVD, that has ISOs, that, you know, has audits on cybersecurity and so on. So it's a different type of product. Marvel might be useful, especially for research, but it's not something that it's ready for clinical application. And then we could maybe do a deeper dive into the performance of Marvel as compared to AION. But they have different use cases from what I can gather. Another question is, we have a detailed knowledge base of variants in our institution. Can we bring it there?
Yeah, definitely. We can both like import like your old files and also like classification so that you don't miss any historical data. So basically we have like transition period in which we provide all the support so that like you can incorporate any previous knowledge.
Great. Two more questions and then I think we can wrap up. We have a complex reporting infrastructure. Can we still use it when using AION?
Yes, I mean, we have done a lot of work in terms of like integrating our platform both like at both ends, like on the one hand side, like to, you know, collect like the FASTQ files if you have a particular, you know, system that in which like you would like to integrate and launch directly the cases in AION and via API, and also for reporting purposes as well. So basically, AION wants to be like the connection in between, like, you know, the lab and the patient. So any integration and work that needs to be done in between, we can support into this. Obviously, like we need to know like the specific requirements for your case, but like yeah, definitely we can work on that.
Okay, and one last question that I can see from here. You keep talking about phenotype prioritization. What is the difference, if any, from filtering by HPO terms? And maybe I can respond to that one. We just received a last minute question. We'll also tackle that one. What is the difference between phenotype prioritization and HPO-based filtering? They're pretty different. They try to get to the same result of, at the end of the day, narrowing down the number of possibilities or the number of variants, and only having to review variants that are compatible with the patient disease, meaning that are possibly pathogenic or causative for that disease. The main difference from a practical perspective is that if you filter by HPO terms, usually implementations go that if you increase the number of HPO terms, you decrease the stringency of the filter. most times. And this basically is counterintuitive because with higher number of HPO terms for the patient, you should be more precise also in the way that that you get to the results. And basically, phenotype prioritization allows you to basically have a process where the higher the specificity of the input phenotype, the better the results, while also allowing for some sort of fuzzy logic where if a term is not exactly the one that you implement that you entered, then it's going to be able to take that into account. Besides that, the algorithm can be, in that case, arbitrarily complex and take into account different correlations between symptoms, etc. So phenotype prioritization, it's way more powerful than filtering by HPO terms. But again, in HPO terms, filtering for simple cases can work, and it depends on the implementation. And the last minute question that we receive is, does it work for long read whole genome sequencing? And I'm sending this to you, Aina.
Sure. I mean, thank you. And the answer is yes. So basically, both with ONT and PacBio data, and actually we have like tested this in different settings and different requirements. Something also like was very nice is like, I think even you, Pablo, can give a bit more insights, like because we have been in this UDNI hackathon in the Netherlands, and in there, like, you know, because there was like a bit more of research purposes for diagnosing, there has been like a lot of different formats, different structure, different kind of data. And definitely we had the opportunity to test like a lot of like long read data. So I think we definitely can confirm that it works for long reads.
Yes, that was a great experience. Okay, I think we need to wrap up. I'm sharing now Aina's email also for you to get in touch, and if you're interested in AION, please get in touch, and we can help you organize a quick testing phase, and then if you're interested, obviously we can engage in further conversations. For now, I just want to thank you again a lot for participating in this webinar, for attending, for all the very interesting questions. They were actually very interesting. And again, if you think AION is a tool that would help you solve interpretation bottlenecks within your institution or help you expand your research abilities also within your institution with a streamlined process that is very homogeneous for many cases, please get in touch and we'll be very happy to help you. So with that, we're concluding this webinar. Again, you have Aina's email address here. You can get in touch also through the website. Thanks a lot. Thank you, Aina, for the demo, for participating, and for helping all of the attendees understand AION better. And thank you also from my side. Have a good day.
Thank you.
Thank you.